began at Winston-Salem State University, where he pursued biology studies. In 2016, he graduated for the Integrative Molecular and Biomedical Science PhD program at Baylor College of Medicine, right? And then received postdoctoral training first at Mayo Clinic in Rochester, Minnesota, and later at Carver College of Medicine at the University of Iowa. Nowadays, Dr. Hinton is an assistant professor at Vanderbilt University's School of Medicine. His research centers on the molecular mechanisms underlying diabetes and cardiovascular diseases, focusing especially on, or specifically on mitochondrial and endoplasmic reticulum protein interactions. Dr. Hinton's work has earned him recognition, including awards for his imaging techniques. He has published extensively and serves on several editorial boards. To date, he has published about 100 papers, receiving more than 50 awards and given over 200 invited talks. Alongside his research, Dr. Hinton is committed to mentorship, having guided over 90 graduate and undergraduate students. He also actively engages with professional societies advocating for diversity and inclusion. Next, I give the floor to Dr. Hinton, thanking him again for joining us today and sharing the progress of his research.
Thank you, everyone, for having me. And sorry that I don't speak perfect Spanish yet. Yo necesito practicar en español más, but only for preguntas. Okay. So today, I'll give a talk focused around my research. It is focused on the MICOS complex. In our laboratory, we focus on electron microscopy. So one of the reasons I'm here is not only to give a talk, but to recruit. So we can all also introduce you to my laboratory and with the hopes of continuing the collaboration. So just to highlight how diverse my lab is, these are the areas that we publish in, and then also some of the areas in diversity, equity, inclusion. So I have a research assistant professor who's here today, Dr. Andrea Marshall, and Dr. Zarbu is a research instructor. I have 3 visiting faculty from different countries, and then Edgar's a staff scientist in the laboratory, Dr. Edgar Lopez, and then also we have several postdocs, and then we have grad students, and also post-bac and research techs, and then a lot of undergrads. So there's a lot. I've been featured in a lot of places. I've had some interactions being on the boards with like Francis Collins for like Scientific Trust. So I have experienced pretty wide experiences, but for my research, I'll be talking about some of the methodologies that we've done to develop around mitochondria and other organelles. We'll be showcasing some of our 3D EM. So that covers FibSim and seroblock facing, which I'll explain later. And then we also have done some work in mitochondrial structural changes in tissue. I'll explain that in more detail, but we have a CZI grant for over $1 million to look at the entire structures that exist in mitochondria across aging in every tissue. So we're also looking for collaborations in like eye, for example, because we don't specialize in the eye, so that's also a reason here. And then we study for mechanistic things. I know you're like, well, you can't run a lab just on structure. Mechanistically, we look at the MICOS complex and OPA1 and its role in regulating mitochondrial structure. You see that our pretty pictures do get us a lot of different covers, which is really nice. We actually have 2 more covers coming out in Journal of Cellular Physiology pretty soon. And then we have some more developed papers around human tissue. They take a lot longer. There's papers in revision at Circulation Research for a long time. And then we do DEI work and mentoring work. So a lot of my work has been featured in Cell, Nature, Science, Lancet, things like that. And we also have a lot of mentorship opportunities. So when you're in the laboratory, you actually get to develop your mentoring strategies. So we focus on holistic mentorship. And we've had a Fulbright winner and also a Marshall Scholar winner in our laboratory. And our laboratory is just, not that large, but it's mighty in what we have. We've had 6 undergraduate winners of a prestigious national conference in various areas. And then we've had 1 actual undergrad that's gone on a medical student, that's a medical student now that actually has a paper that's been cited over 100 times. It was 80 at the time this poster was done. So you can see that we pride ourselves in focusing on undergrads, graduate students, and postdocs because every stage is important. And these are some of my collaborators. And this is my funding, which we'll get through. So in my lab, we focus on 4 areas. So you heard in the introduction about insulin action mechanisms. Today, I won't be talking about that. We also have an area that's under development, and also why I'm seeking another collaboration around brain research, focusing on more blood pressure regulation and the MICOS complex. Today, I'll talk a lot about mitochondrial dysfunction and then method building to kind of give you a high-level understanding of what we do in the laboratory. So today, when I'm talking about mitochondrial dysfunction, it's based off of 2 projects that are funded. So we have a CZI project from Chan Zuckerberg Institute that looks at mitochondrial structural changes in humans, cell lines like myotubes, mouse, and fly. And for the humans, we actually have 2 IRBs that are active where we take autopsy cases. So if that's something of interest to you, you can do that with Dr. Marshall, who does a lot of those particular things. If you're interested in just doing the EM and structural stuff, you can do that with Dr. Lopez. And then in particular, the AMIRA and 3D-EM workflows are something that Dr. Lopez developed, and then the workflows for taking patient samples is what Dr. Marshall's developed. We also use Python and MATLAB in the lab, but you also can do manual segmentation if you're not familiar with that. But if you're up to learning, we're happy to teach you. We also have another project funded by Brio's Wellcome Fund and also the United Negro College Fund and Bristol Myers Squibb to focus on molecular mechanisms of action that can help refold cristae. So I'll just show you a quick slide on that we've started to do with a pharmaceutical company, which is really fun. And we're building this database that we hope to publish and sell one day around all tissues that show different aging structural changes. The reason this is important is because with pathologies, it needs to kind of continually be updated. Right now, when you're a pathologist, you do EM slices and HE staining, and sometimes when you're surveying for mitochondria or other organelle defects, what happens is you miss sometimes the percentage of cristae that are altered or the structures that are changed. Usually around 25 to 30% of the EM slices have defects, but it's by chance that you may not catch that. And with 3D EM, you're able to catch that as well as the integrated structure of the cristae as well. So all of us are familiar with this Hallmarks of Aging paper. I'm only really care about mitochondrial dysfunction and epigenetics. I think everything is, nice to study, but we Of course, the parts of the mitochondria is in a cell. You see cristae, which are these invaginations in the matrix, and an inner and outer membrane that help participate in fusion and fission. But we recently wrote a paper about the different structures that are observed with mitochondria in the tissue. We're not the ones that found all the structures. It's a pool from all the literature and surmising it together so that everyone can understand what's available to the community. So currently, there are 8 shapes of mitochondria, and there may be a 9th shape that was just published recently by Jonathan Friedman, who's at UT Southwestern. So it's really nice. So the mitochondria field is ever evolving. So just to highlight a couple, megamitochondria are naturally found in liver. So sometimes when you've looked at maybe EM images, you see these very large mitochondria. So that's a megamitochondria. But they also exist in disease such as cancer. And then mitochondria work really in unison to help you flex and lift your muscles. And that is a lot with branching, and that's where the distribution of ATP happens a lot. And then there's these nanotunnel structures where you have defects in mitochondrial metabolism or a gene change or sometimes mutations in mtDNA, and it leads to this rare shape where there's void of cristae in the middle, and then there's mitochondria that are sick on both sides. So it acts as an incomplete fission mechanism. So just for you to kind of know that mitochondria are a lot more complicated than the kidney bean textbook image that you see. There are a lot of different unique shapes of mitochondria, and it's inside not only are the shapes different, but the cristae are as well. So there are onion-shaped, lamellar-shaped, and tubular cristae, and then there's also dynamics that happens with mitochondria, but also the cristae. So we actually know that mitochondria exist as a large shape, and then they can divide with fission into two mitochondria and glue back together with fusion. But interestingly enough, cristae have their own mechanisms recently discovered in an EMBO paper in 2020. So now cristae also fuse and fizz. They do transverse, X-type fission, and Y-type fission. And this is very understudied in an area that everyone is kind of chasing after to figure out what this may be specifically in each type of mechanism. So there's also fusion and fission mechanisms that join the mitochondria together and separate. And there's also other organelles that come in contact with these mitochondria, like ER contacts. And I wrote in this review about how we need to kind of think about changing how we study mitochondria in the field. One of those highlights is looking at the last three, looking at nucleoid distribution, where we're also thinking about a skeletal muscle collaboration, where we're focusing around maybe how satellite cells generate nucleoid distribution differently based upon how much time they have in nutrient signaling, or if they're starved, or different things like that. And then also we looked at tissue-dependent effects, which is what you'll see today. And then lastly, we'll look at mitochondrial phenotype frequency and understanding that the frequency of mitochondria per tissue is varied based upon disease or a natural tissue. And we're trying to figure out if we can push the diseased mitochondrial structures back to the normal phenotype structure. And so I'll show a brief part about that in the heart failure paper. So I talked to you all about a mitochondrial connectome. So of course I'm talking about kidney, liver, and gastrocnemius, and soleus, and cardiac. But one of the projects I'm not talking about is the sperm. So if you come to the lab, you'll be able to take sperm samples from humans, actually do 3D reconstruction of them, and then be able to actually knock down a gene of interest that we study and look at the motility and see how it changes, because mitochondria are quite important for sperm. This doesn't feature the head of the sperm, and we're also looking at Leydig cells and also germ cells as well. And we have an interest in brown adipose tissue as well. We've published 2 smaller papers, and now we're working on a much more mechanistic, larger paper as well. So this is just a high level of some of the things I'll talk about. So I will talk about some work that's published in AJP Heart, looking at aging mitochondria structural changes, and then also this aging cell paper that features more skeletal muscle. And then I will lastly talk about a method that we've developed around 3D reconstruction that I just want to highlight, that I'd love for you to read. And if you're invested in EM, this is something that I'd love for you to add to your projects. So I won't spend time on this, but this is something that Dr. Marshall and Amber, a graduate student in the lab that's close to finishing. She developed a cristae score developed around 3D reconstruction. And so what we can actually now do is determine, based upon a certain level of cristae information, how to designate if the cristae are whole or not whole. The reason this is important is because there's one in 2D, but there's never been one developed in 3D. So this is why the paper got the cover and the importance of it, not just because of the tissue source that it was coming from. We use adipose tissue because the cristae appear very clean, and that's why we did that method. But you can basically see that we can use 3 different measurements, number of values per type of instance between young and old, population's frequency changes of cristae score, and then also looking at the cristae score phenotypically. So we quantified this based upon phenotype, and now this is a method that can be used. So we also are looking at the human connectome. It's a little bit slower because we have to have people donate things, or people have to, pass away, and we respect their bodies, and we're able to work with the families to take those tissues to find out what happened. So what I have up here is part of the skeletal muscle paper that we're actually sending back into Aging Cell in a second. It'll be on Wednesday. But basically, we take MRI images, and we also do 3D reconstruction of human tissue as well. And we've done it in the heart and skeletal muscle, and we're now starting to collect cases for other tissues as well. This is a 5-year project for CZI, so it takes some time, but the mouse project will be complete pretty soon. I'll feature some of Dr. Zervu and Dr. Peter Ajay's work on heart failure structural changes, and we now have a mechanism that we're searching in the 3D reconstruction space. And I want to highlight two more things. I would love for you all to stay tuned for this paper to come out. I think this is a fabulous work where we've done across continents. So we do a lot of collaboration across continents. So this is why I'm bringing this up. So we have a postdoc from Brazil, a faculty member from India, and an individual from America, but she also is Hmong, so she came from Southeast Asia as well. So we love collaboration in the laboratory. We think it's the best thing that we can do. So we also like to share collaboration by doing co-first. So actually more people get a lot more papers in the laboratory because one person will be primary designated, but they'll have co-first to help them, and they'll flip back and forth between. So in the event of maybe 2 years, you can have 3 first authors and a couple co-authors versus having one primary first author. So we think that strategy is much more effective. Why is that the case? From a faculty job standpoint, we already have 3 postdocs that already had faculty jobs more recently this past year. So we think this method is highly important and something that you could feature if you come to the lab. You see I'm heavily trying to recruit. And so the last thing is we believe in undergrads doing the same model. We love to pair undergrads from different backgrounds so that they can work together and figure out their commonalities that they have, and they can drive the research forward. So you can phenotypically see that everyone's different, and but very much the same stage. They're all undergrads. And with this work, it's been phenomenal. Brianna has gone on to have 2 summer programs, and now she's on her 3rd. Ashton just got into a PhD program, and he wants to come back to the lab because I'm a faculty at Meharry and Vanderbilt, so he can come back from Meharry to the lab. And then Mason is almost done with his undergraduate degree. He's actually a post-bac, but also undergrad at the same time. So he comes back every winter, every summer, and any other break that he has. So we list him as a post-bac, and he does conduct research in the laboratory. And this is on human skeletal muscle, looking at another gene that interacts with the MICOS complex. So let's kind of get into the methods of everything. So basically today I'll show you a paper around heart muscle changes in male mice, skeletal muscle changes in male mice, and male mice for human hearts. And then the hope, when we get a little bit more funding for this, to compare and look at male versus females. And so the reason that we're only using 3 mice is because the imaging technique that I spend money on costs about $14,000 for 5 to 7 samples. Yes, that face said it all. or MATLAB, so we gave equations specifically for MATLAB so that if people get the software we have, it's like a cookbook. You can just follow it and put it in process. And then we also showed how you can do things with manual segmentation in a way that's still fast, even though you have to cover a lot of space. So we showed that you can either do 50 mitochondria across 8 mice to get the same result, or you can do 250 mitochondria in a more deeper analysis across 3 to 5 mice. And so that's what we've done. So this is our method on how we develop this. Then we have assigned other organelle structures. So there's not only mitochondria in a cell, so it's important to characterize those correctly and quantify those as well. So we've done that with a recycling machinery or autophagy machinery. And then we've looked at mitochondrial fixation. So I want to go here. So I want you all to think about how you fix mitochondria because it actually varies in shape. So one of the things that we've learned is that if you're doing metabolism or mitochondrial changes, you should definitely consider following our guide. If you're not so much focused on that, okay, maybe not, right? But to kind of introduce this to you is that when you use CO2 for mice, you actually create an artifact that's a nanotunnel, that structure that I showed you earlier in 3D, versus more of a fluid kind of structure if you're using isoflurane. And then for anesthesia, specifically just for mitochondrial metabolism, if you're doing PBS and PFA, you create this artifact versus we're just using only PFA by itself. And if you're going to do immersion fixation where you're cutting off a small piece of tissue and putting it in fixative, you want to do a small piece, 1 millimeter cubed or smaller, so that you can actually have proper structural change. Because a lot of times when you have large pieces, they form these artifacts too. And we chose brain just as a random tissue that's totally different than what we've done. And we also show that these same things work in skeletal muscle, which is what made the cover. So this is something that Prasanna and I did together. And to introduce you to some of these other organelles that we've been talking about at the high level, you can see this image. This is a FibSim image. So it's focused ion beam scanning electron microscopy, and I'll explain even in more detail later. But this is what you're capturing as a MERX, and you could study MERX in the laboratory. So this white space that's connected to the red, that's mitochondria, and ER, which is green, it's where the connection for these proteins are housed that connect the mitochondria to ER. And it can also be connected through actin interactions, lysosomes, and lipid droplets as well. So they all are studied in different ways when we have alterations in protein structure. So let's kind of get into the microscopy and let's get into some results now that I've explained how the lab works, tried to recruit you. So now I'll try to impress you with some of the stories. So we look at visualization in 3D. One of the ways that we do this is with serial block facing. So we call this SBF, SEM. So the reason this is important is because what happens is we take a diamond knife to go across the epoxy resin, and then once the image is completely cut off, then we scan it, and then it's uploaded. And then we do this in serial sections, up to 1,000 slices, depending on how thick the tissue is and what information you need. And then you can reconstruct it here using a mirror software, manual segmentation, or AI learning, which is the Python-based things. Then we've done FibSim, so we've featured several times in Trends in Biochemical Sciences around the techniques that we do. So we have a story around CLIM, which I'm not showing, which is a technique that we're emerging to do. That's, yes? Yeah. Structures. We've developed several ways to do the structures. So we do volume, perimeter, and area. This is what the Amirasoft will spit out. But you also can do barycenter, which describes objects in physics like planets, if you will, from a certain point to measure to others. You also can look at mitochondrial length. You also can look at circularity index, which is an equation based off of volume, area, and form factor. Then you can also look at mitochondrial complexity index as well. So we use each of these calculations to not only phenotypically look at things, but quantify. So each of our papers features all of those measurements. And then we also, to make sure that people see our mitochondrial structures, but not biasly, not only looking at the phenotype. When I had introduced this paper, you'll see here, one dot represents 250 mitochondria per mouse, and then you actually see the error changes of each mitochondria as well. I'll zoom in on some of this so that you can see a lot later, but we also do this per tissue as well. So anything that we're monitoring, we do the high level where we aggregate the data together, and then we also separate the data as well. And then to confirm, we not only look at things from a structural standpoint because it's static, we also look at confocal to look at dynamics, seahorse usually, gene expression by qPCR changes in Western blot, and then we also independently usually have another lab kind of reproduce some of our results. So this is kind of the standard of how we quantify. So great question. Yeah, image J. Because that's so for this particular thing, once you have the FibSim, you can also do image J or mirror software. So we get a lot of data. The first thing you actually have to do is like aggregate the slices, and then you actually have to put a filter to make everything grayscaled if you're going to do the automated segmentation. So for FibSim, you can use AI-based things and be able to generate structures, but manual segmentation is more likely for serial block facing. There is software that's available called MitoNet that is in a Cell Systems paper that Kadar developed that's at the NIH. He's part of the NCI Institute. He developed a way for you to be able to quantify automatically as well. So that's more of a feature that's more for FibSim images, but it's on the market for doing serial block facing. the 3 papers that we're going to talk about in much more detail. So we're going to look at these 3 papers and then a heart failure paper that's in revision. So I just want to tell you that you also can train very nice undergraduates in the laboratory. So Kid is a student from Hawaii, and he actually just won the Marshall Fellowship not too long ago, just a couple months ago. And he's a co-first on each of those papers that we listed because he actually wrote the paper. He understands the paper very well. We treat him like a colleague. So it allows for individuals to kind of immerse themselves in a lot of well-roundedness. So he was an undergrad, then did a post-bac, and he has about 30 papers in the laboratory. So at a high level, we're looking phenotypically, but a second we will look at quantification, and I'll explain more of the details what Edgar's talking about. So phenotypically, it should be very easy to point out differences if there are any based upon just looking at structure. So we have 3-month and 2-year muscles, and also in soleus and gastrocnemius to be specific, and cardiac. And from this, we can compare the 3-month tissue to the 2-year tissue. And you can see that the mitochondria are a lot more joint together in 3-month for the soleus as well, and then also the cardiac. But in the 2-year, they're a lot more fragmented. So now the question became earlier about how do you introduce quantification in this, because that's the most important thing. You're looking at phenotypes, but it really doesn't mean anything unless you can quantify and then link it to a phenotype back to what could be either a mechanism or a disease. So one reason that we focused on this is because we can quantify in multiple ways. So we can use volume, area, and then also perimeter. So one of the things that the software spits out is a 3D EM version of area and perimeter. The reason that it does that is because it uses those 2 parameters to actually get volume, but also other equations. So for example, you can plug in MATLAB and you can use a calculus-based equation that's based off of a square, and it looks at a perfect circle and it assigns it 1. And so that's the circularity index, or sphericity in the case of the 3D software. But then if it changes, it becomes a polygon or some other type of shape, it becomes less than 1. And so each time a mitochondria is read by a different color, so that's why there's different colors, and you can assign a value to that. And so we call that a material in the particular software. And then you can also do mitochondrial complexity index, which is actually based off of confocal calculation. It's called shape description or form factor. So it allows for you to see the shape of the mitochondria. We extrapolate that into 3D. It was proven by multiple labs before we got to it. Martin Picard is the one that brought that to the field, going from 2D to 3D. That was done in a Cell Reports paper. Martin Picard is at Columbia. He's one of the big Olympian gods in the mitochondria field. Is like I like to tell the story in my laboratory so people understand who's important in each field. And so not only doing this is important, but we also can show the distribution of total volume. So it's important to see that the blocks are equal as far as total volume, but the way that the mitochondria are arranged, it makes for fragmentation a lot more easier to visualize in the older samples. So why would that be important? It's because you can actually karyotype the mitochondria. So everyone's familiar with chromosomes and karyotyping. We call this mitotyping, where you actually can look at things by volume. And so what you do here, you can look at 3 months and 2 years, and you can characterize all the mitochondrial structures. This is a snapshot. Obviously, this is not 750 mitochondria, but it goes along from all the mitochondria that I measured and look at the largest to the smallest. And so this gives you a lot more visualization to be able to see the changes that we observe by quantification as well. And you can see this per tissue type. And so in the soleus, they're a lot more organized, even at the 3 to 2 year comparison, but they're less, you can see. At 2 years, you can see they're more fragmented around this area, but overall, they're very similar. And 3 months to 2 years in the cardiac, there's hardly any changes. And I want to point that out. That's important. I do think it's really important to remember that there are changes in the levels of mtDNA or mitochondrial DNA per mutations, even when you're getting older. And the cardiac tissue is much more resistant. So without a challenge, you really shouldn't see much of a change. At first, I really thought my hypothesis was right. I was like, we're going to see a lot of fragmentation. The whole lab laughed at me because there was no change. So I said we have to kind of really probably hijack the system by doing heart failure changes. And so that's where we got into that 4th paper around this concept of muscle in the laboratory to see these challenges. So when we quantify this, we can use mitochondrial complexity index or sphericity. And these are 2 additional measurements that look at the individual structures and actually observe the unique shapes that they are. And so what we can see is that with complexity index, there's less complexity at 2 years and more sphericity at 2 years, which suggests that they're getting smaller. So they should go in the opposite direction when they're equivalent. If they go in the same direction, then they're not equivalent. So something's actually kind of wrong with either your equation, or there's actually some really skewed mitochondria that you're seeing in the system that would actually make this change. But both should be reported, just to let you know. And then now, we always think about mitochondrial structure as very fascinating, but one of the things that we need to do is look at the MICOS complex or another mitochondrial shaping factor to observe if we can link the structure to mechanism. So one of the things that we care about in the laboratory is looking at cristae. So the MICOS complex, this organizational complex, is made up of 7 members in the cristae junction. So each one of them are assigned MIC, so that's referring back to the MICOS complex, and a different kilodalton value. So they are different in each system. So in mammalian systems, they're called MIC60 is the largest. Then there's MIC19, MIC25, MIC10, 12/13 if it's in human or mouse. And then there's also MIC10. And sometimes there is other complexes that are 27 or 26. And sometimes they exist as pairs. So like in flies, you have less MICOS components because they're similar homologs. But in humans, you have all the components. So looking at this, we've noticed that MIC10 helps stabilize ATP synthase. So that was a paper that was recently discovered where the monomeric forms of MICOS stabilizes the complex and allows for ATP synthesis to occur much more efficiently. And the cristae junction is important because it keeps it tight for communication with TOM, which allows for mitochondrial transport into the mitochondria. SAM complex, which helps form beta barrels and shuffles into the complex. And also MIA14, which 40 that participates in the same thing. And then also it interacts with OPA1. OPA1 is a really important protein that maybe some of y'all have heard about in the eye. It was found there. It's called optic atrophy 1. And this particular protein actually forms the cristae. And it's the master regulator of this. But now we're forming new hypothesis around that, the idea that there's some helper proteins that OPA1 uses to be able to form these cristae structures. And one of those is the MICOS complex. So when looking at the MICOS complex, we know that it exists in the cristae junction, and we know it's really important. And this paper that came out in 2015 showed that OPA1 in purple interacts with CHCD3, or MIC19, and MF, which is mitophilin. And it forms this complex to regulate cristae architecture. So really, OPA1 has some helpers, if you will. So think of OPA1 as Santa, and it has many elves that are running around in the system to help regulate all the toys that need to be transferred everywhere. But when something's not communicated correctly or the message is not translated appropriately, it goes from this beautiful lamellar cristae structures to these tubular cristae structures that I showed earlier. So this is why there's different phenotypical changes that occur. So when you decrease the levels of OPA1 protein or MICOS complex proteins or at the gene level, you see alterations in the cristae structure and you see these tubular shapes. So what we decided to do was actually do a screening of all the tissues. So all the tissues see changes in MICOS, but a variation. So I'm just focused on just skeletal and cardiac. So in skeletal muscle, we focus on gastrocnemius in mice and cardiac tissue in mice as well. And we saw that there were MICOS changes. So just to introduce you, OPA1 is the kind of positive control because we know that with aging it decreases, but the hypothesis is around understanding what happens to gene expression with the others. And when we look at this, we find that there's a decrease in gene expression in MICOS complex in mice at 2 years and also cardiac. But what we did want to see was if this happened in Drosophila as well. We have fly models in the laboratory as well. So what we look at is an evolutionary evolved fly. We published a small paper in IGMS showing that this particular fly model allows for us to have metabolic changes in both male and female, but also has the idea of transcriptional changes as well, some more pronounced in males than females and vice versa. And we also saw changes in gene expression of certain mitochondrial genes. So when we did this, we actually looked at MICOS complex, and we saw that in skeletal, there's only really MIC19 and MIC13, or QIL1 it's called, that actually changes, versus in the MIC category, MIC60 changes in the heart and nothing else changes. So this is an evolutionary evolved fly. So now we're looking at kind of expanding the study to knock out the genes of interest, particularly in thorax or skeletal muscle or flight muscle, if you will, or the heart tube or in the brain. So to functionally look at this, we always like to look at cell culture. I think it's such a powerful system. So to make myotubes, you isolate satellite cells from skeletal muscle. And then you actually allow for growth media to hit the cells. They germinate and they'll make myoblasts, then they'll go to myocytes, and they'll differentiate into myotubes 4 to 7 days after the other process. So each stage, you want to be able to think about like about 2 weeks in time before you can actually do an experiment. And because they're mitochondrial-derived genes, you actually have to wait till they're fully formed into myotubes before you do the gene ablation or the transient CRISPR change, because if you don't, it affects metabolism. So this is why we chose CRISPR to be able to look at these particular knockouts. So we knocked out CHCD3 and CHCD6, and we also try to highlight that in a human or in a fly, they're very interchangeable. So by color on Kiko, you'll see that there's 2 different genes, but they're also very similar. So we just wanted to highlight that in the paper. So basically, in mitochondrial area, you see a decrease in overall mitochondrial structural changes. It's very clear to see that you see a variety of shapes of mitochondria, but in each of these, you see alterations in cristae morphology, and then you see this where circularity index increases, so that's the same thing as sphericity, if you will, and we see an increase in number of mitochondria. And then we did cristae scoring, where you can observe how many cristae are in mitochondria. Then we did 3D, which is our classic technique that we look in the laboratory, and what we find is that there are alterations in mitochondrial structure. Each one of them are unique, but it seems that CHCD6 has the most robust mitochondrial fragmentation. So with that being said, we also wanted to look at another cell type. So remember that there's skeletal muscle, there's fibroblasts in skeletal muscle, so we looked at fibroblasts, and we also looked at cardiac fibroblasts too. And in both cases, the fibroblasts were altered, so we used Imaris to quantify this, and we looked at the ratio between tubular and fragmented mitochondria. And we observed that OPA1 had a huge decrease, and then the MICOS complex had variations in the degree of fragmentation compared to the control. So then we did seahorse. So if you're not familiar with seahorse is not looking at little beautiful seahorses that are in the sea, but it's a machine that looks at how respiration changes. So one of the things that it commonly reads out is OCAR, which is oxygen consumption. Oxygen consumption is very important because it allows for you to measure different states of the electron transport chain. And if you back up, if you remember, the MICOS and OPA1 help propagate the electron transport chain. You saw the complex at the bottom. So the tighter the cristae, the more effective the electron transport chain occurs. That's why there's sometimes this variation in the amount of ATP that's produced. And so we want to be able to do a readout of this. So steps 1 to 4 is basal consumption. Steps 5, 6, and 7 use a drug to block actual ATP synthase. It's called a ligamycin. And then steps 7 going into 8, which is 7, 8, and 9, use FCCP to push all protons into the system and give a maximum burst. So that's why we call maximum consumption. And then lastly, you use 11, 12, and 13. And that step, you use 2 drugs, one to block complex 3 and the other to block complex 1. And then that's with ronanone and entomycin A. And then you can do this readout. I know some of you are experts in mitochondria, so we don't use non-mitochondrial or proton leak. We're saving that for another paper. We thought that was an interesting phenotype, just to tell you. So basically, at each of these steps, we use a positive control for OPA1 throughout all the papers. And then we look at the MICOS consumption changes after ablation, and we see that with basal, it's decreased compared to the control. ATP link is decreased, but it's a lot more variation in the MICOS complex. Maximum capacity, they're all decreased, and then spare capacity, there's not as much of effect in mitophilin, but the others, there's a huge effect. So with that, we wanted to do a snapshot of metabolomics. So this gets into some of the screening that we did for drugs, which I'll tell you about where we got some funding from pharmaceutical companies. So this is like, why are you looking at all this? There's no need to kind of explain everything except for this particular hit here. So when you knock out mitophilin, you do see an enrichment for multiple pathways, but one of the cool pathways is sterogenesis. And the reason that's important is because you see cholesterol hits, testosterone, estrogen hits. And so my previous mentor, Dale, who's in National Academy, he discovered this whole process. So it's a paper under Nature Metabolism and Review. You see the EM? It's nice EM. I did the EM. But the first author is Dr. Rhonda Sumner, who's at UCLA. But basically, what they did was they used wild-type mice, and they isolated brown adipose tissue mitochondria. If you see a theme, mitochondria from brown adipose tissue is so pretty to see. And then when you knock out a gene like OPA1, you actually have no cristae anymore. But then when you actually give estrogen, you can refold these cristae. So I recently got a grant around this and understanding what are the new mechanisms that may be associated with the MICOS complex. So one of the things that we're actually doing is knocking out the MICOS complex and refolding cristae, but not just to look at it phenotypically. We're looking at the mechanism, looking at Raman spectroscopy to look at the cardiolipin and see how it refolds with the hydroxyl groups. If it does use the hydroxyl groups, it may use something else. We don't know. And actually see mechanistically how that changes over time, whether it's with estrogen or testosterone, which is more flexible. Is it because there's two hydroxyl groups with the estrogen, or maybe it may be a serum mechanism, serum mimic like a tamoxifen, maybe also have really good refolding. So it's something that we're investigating right now, and we need a postdoc to be able to help to do this particular project, just to throw that out there again. So we also looked at CHCD3 and CHCD6. You already saw at a high level what happens. So when we look at this particular part of enrichment, we do see changes in amino acids and starch metabolism. So what we decided to do was go back to the tissue source to see if we could actually see in whole tissue a similar phenotype. So I'm not going to get into the specifics because metabolomics is a lot. You can read the paper. But the high level is we did recapitulate the phenotype where we saw amino acid changes, NAD metabolism changes, and also lipid changes. So one of the things that we did show was NAD metabolism changes, because in aged tissues, you should see a change in NAD metabolism. So if you're not seeing that, that's not a good positive control. But the thing that was novel from this part of the study was cardiolipins. And this is why we decided to pursue the mechanistic studies with the NMR, and then also looking at Raman spectroscopy, which is really cool. And we're hoping to do that with Vermont and then also at Vandy as well. So if you also are interested in lipidomics or metabolomics, we have those capabilities. We're happy to do that. My lab is one part of a bigger lab that also Melanie McReynolds, who's at Penn State, does most of the metabolomics and lipidomics, and we combine our lab's expertise to make a lot of papers in this aging series that we're doing. So I told you that my hypothesis was wrong, and my hypothesis was wrong for this paper too. I thought there was going to be fragmentation, but there's a lot of fusion. So I actually want to highlight this to you. So when we look at normal hearts, and this is funding by the pharmaceutical company and also the United Negro College Fund, they give a good amount of money. So it's like $470,000. We received 2 of those grants. And so basically the normal heart, you've kind of already been hearing about the description of, normal ultrastructure, normal dynamics, efficient ATP synthesis. But in failing hearts, we actually see alterations in structure and fibers. We see alterations in paired dynamics and decrease in ATP synthesis. So we think that structure is actually directly affecting function. But this is something that's just a hypothesis. We have to see if that's the case. So what we did was we used BioView, which is a database that we have at Vanderbilt, where we sequence up to, right now it has a total of 1,000, but for this particular part, 85,000 was all that would work for our particular part. But you receive an ICD code from the hospital saying somebody has heart failure, someone has diabetes if you want, and then we can look at that data and compare that gene expression profile that we see compared to an RNA-seq reference database that's at the Broad Institute, and using algorithms that are proprietary to only Nancy Cox and Leah Davis, who are our collaborators, you actually can look at phenotypic responses. And when you see this, you can actually assign genes and see if they're associated with heart failure. So either we'll look at nominal significance, which is less than 0.05, and remember, we're only looking at mutations, so these SNP changes, okay? We're not looking at like fragmentation, breaks in the entire gene or the chromosome. And then we associated this with heart failure. And what we found was that it was very interesting. Not many genes show changes with ethnicity. And so we were able to describe that European individuals had odds ratios for these SNPs that were increased. And then in African American population, we saw that was decreased. Now you're going to have the interesting question. I know you all have seen the Nature paper that came out around Latino, Latina, and Hispanic, like actual information. So our database is underpowered, just in case you're thinking about the actual specifics, because it's an admixture. So we do see some people they'll say that they're only European, or they'll say they're only Black, but then you can see that there's some like segregation to like certain Latino gene identifiers, right, or Latino gene identifiers. So it's very complicated when we get to that part because it's self-reported. So we're not phenotypically looking at the genome for those particular responses. So we can't really code for that. So we're underpowered already. for Black people at 12,000 patients, and then for individuals that are from Latino backgrounds and Asian backgrounds is even significantly lower. So we have to use other databases to be able to do that. There is a Japanese database that actually serves as a reference. And so people are now trying to build up these databases. So it'll be maybe 5 to 10 years before we can get more clearer pictures about everything. So I just wanted to throw that out there. Not including anybody. It's just that we're just understudied. But that is a grant that we could put in for a postdoc to study that. So just letting you know. And then what we did was we actually took autopsy cases that Dr. Marshall collected from Iowa and then also at Vanderbilt. So you get to travel a lot. And basically, we had 4 accident cases that passed away and 4 heart failure patients. So we collaborate with Brian to do the 3D for FibSim. He's at the NIH, so he's the leading expert in the field for fibers and structural changes. And Olu is a cardiologist that allows us to get access to human tissues when there are rare cases that we don't have at Vanderbilt. So UCLA sees so many patients because it's the largest medical system in the US, and so they get a lot more tissues banked, and so we're able to access that. So when we do this, we wanted to look at the signature of disease. There should be some baseline phenotypes that are associated with heart failure, and one of those should be NAD metabolism changes, branched chain amino acids, and also looking at glucose metabolism changes. So because we see those, we feel comfortable moving forward with the project. So then what we did was we did 3D reconstruction on 4 patients each, 4 controls, if you will, and 4 heart failure patients. And what we found, which was, I was not expecting this, was that the mitochondria are a lot larger. So they're fused, and I have no clue why. So it was something that stumped us for a while. And then we asked the question, well, maybe the fibers are contributing to this, because you can see how unorganized the fibers are in heart failure. So when we looked at the mitochondria by mitotyping, we did see larger fused mitochondria, so the sphericities decrease. But then what we did was we looked at the fibers to look at the arrangement. So new data actually describes myofibrils as multiply connected, not so much individual as what it shows in the textbook. This is something that Brian previously did. So this technique is grafted onto our study, where he's co-singer with me, where he's developed this workflow in iLastic to be able to actually look at these fibers and see where they're connected and where they're separate. And so when he does this, he can actually see that in nice normal heart tissue, you have these fibers very well organized, but the position changes in heart failure. Not much, but just enough to alter the change in structure. And so you can see really up close with the heart failure case that the fibers are not as organized as what you see in the control. So what we decided to do was go back to the MICOS complex and look in skeletal muscle and heart to determine if we could see these fiber changes. So we knocked them out. We started to see slight changes, and we said maybe we should make these separate papers. So we just reported the mitochondrial part and the aging cell paper, and we have an MFN2 paper on fiber changes, and now we're working on the heart failure fiber changes, where we actually decided to only focus on CHCD3, which is CHCD6. So based upon how actin is organized, it's different in this particular one. So what we did was we knocked out iPS cardiomyocytes, and we actually looked at the organization. They're immature cardiomyocytes, I just want to be clear. So it could be that the organization could be slightly different and mature, so just to throw that out there. There's always, not perfect data, but you can see that there's mitochondrial fragmentation, the mitochondria are not as large, and that the fibers are not as organized. So we think that CHCD6 may be linked to this and inducing some ER stress. So we decided to test if there was oxidative stress changes. What we found was that there was a slight decrease in membrane potential and an increase in oxidative stress by DCFDA and mitosocs. And so this led us to develop a hypothesis around, well, if there is an ER stress change, what is it doing, right? So we know that when you knock out OPA1, you see an increase in ATF4. So we decided to just pursue the same phenotype with the MICOS. And what we decided to do was knock out ATF4 and use the fly model. And what we found were hits for MICOS, MERX, ER stress, and UPR, and autophagy. So all these genes is a lot of different information, but we decided just to focus on ATF4 because it was such a fabulous change. And then from there, we knocked out ATF4 and overexpressed it. And what we find is that you have fragmentation of ATF4, and then with overexpression, you have these fused mitochondria. So we think that maybe the level change in ATF4 is contributing to the hyperfusion that you see in the heart failure. You're like, this is just in the fly. This is one like image per confocal. It could be anything. So we did the block as well. And we did the entire block this time to look at FibSim. And we actually see that there's complete structural changes across the board. And you can see, if you look at the contraction axis and flip it, you actually can see the same consistent changes. And just for you to see, most of the time I don't show the video for the entire block. I just kind of show what we've segmented. But it's highly important to be able to show what happens through the entire block so that you can kind of believe this particular set of data. It's better to see by eye so you can see all the changes. So there's a lot of lipid droplet changes in the overexpression. There's ER changes and nuclear changes. So the pink is the nucleus, or the magenta. Blue is the mitochondria, and the ER is green. So there's different arrangements, as you can see. The ATF4 knockout is a lot more slices because the mitochondria are so thin, so just be patient. It's turning, it's a little slow. But what you can see is that there's nanotunnels in this particular ATF4 knockout. So now we're pursuing another project that could be for a postdoc at nucleoid distribution and looking at changes in overall structural changes. But the reason this is important for this heart failure paper is because we can see clearly that with the overexpression, there's these hyperfused mitochondria. So it's clear to us that when you're knocking out MICOS, there's this ER stress change that's resulting in ATF4 transcriptional changes that's leading to these huge fused mitochondria. And when you knock out ATF4, as long as there's a little bit left in the system, you have these unique nanotunnel structures. And if so often, there may be a wild type nanotunnel that happens. And you can see how they're structurally different. So we also did this in cells just to make sure in skeletal muscle that we're beating a dead horse and making sure that we make the point very clear. So we also knocked out and overexpressed, and we do see these hyperfused mitochondria and these smaller mitochondria in ATF4 knockout compared to the control. And we do our customary volume, area, and perimeter. So we're in the process of finishing this particular paper. And then we did our classic mitotyping, where you can see some of the larger changes. And then now we're pursuing changes in MERCs, where we're looking at these ER contacts. And now I want to just give you a preview of something that it'll come out like in a couple of weeks on bioarchives. So I added kidney and liver because I was just excited about the data. I've been looking at the data for the past week and a half. So for the talk purpose, like just so that you know what we concluded, there was gene expression changes in CHCD6, which is a MICOS member, in European and African American ancestry. There's alterations in mitochondrial structure in each of the regions, but we just showed IMF. And there's a lot of hyperfused mitochondria. There are some fragmented and branched, but they're all uniquely positioned in a heart failure study to be more larger. When you knock it out in iPS cardiomyocytes and also skeletal and fibroblasts, the MICOS complex, you see mitochondrial fragmentation. And then when you look at iPS cardiomyocytes, you do see changes in ROS expression and membrane potential. Okay, now the preliminary data. That's the cool part. So we also have brain. If anybody wants to collaborate, you could do that under Dr. Marshall. But for this particular part, we looked at other gene expression changes in kidney and liver. So these slides are kind of fast, so y'all can ask questions. But basically, we structurally looked at the kidney, and we see phenotypic changes in the kidney, which you kind of see this classic thing. And then we looked at mitotyping. You can see that the kidney is a lot more positioned to have very unique mitochondrial structures. And then we're in progress to look at MERCs right now for the paper, looking at ER contacts. We noticed that with this particular tissue, that there's ER contact changes with age, which was unique, something that's not as clear in the other tissues. And we're also looking at cristae morphology. So you can see AI is not perfect, so you have to go back and manual segment some parts. And then we've also phenotypically looked at the kidney. So we do see this classic branch chain amino acid metabolism, and NAD metabolism with young versus aged kidney. And then we've looked at these by just showing bar graphs of pulling out some of the enriched coordinates. And we do see changes in lipids, not as drastic changes as what we saw in skeletal and heart. And in HEK cells, we've knocked out the MICOS complex, and we do see these changes in structure, again, but not as drastic as what you see in skeletal or heart. But what's really cool is that we also recently got this ROS data from looking at HEK cells, giving a ROS inhibitor, giving a complex inhibitor and then giving ROS changes because we know that's a known readout. And then we knocked out the MICOS complex, and we saw that there was a decrease in MYC60. So we also collaborated with Dehindra Tomar, who's at Wake Forest, and we did other assays for ROS. And so this is like extra new data. This came last night. I was like, where's this data? So I can show it. So it's really cool. So we do ROS-based things as well. So it'd be really fun for you all to get into this. So we've knocked out CHCD6 and MYC60 in HEK cells, and we compared it to wild type. We did bright red, deep, which is a stain that looks for changes in ROS, and DCFDA, which I've kind of already featured. And so we do see a change in ROS with DCFDA in both. And then with the bright red, we only see a change in CHCD6. And then we also looked at mito orange. I think it's so important to look at the sensitivity of different ROS assays. One ROS assay is not sufficient enough for you to be able to conclude anything, in my opinion. So we also looked at these changes, and we see a change in MYC60, but not in CHCD6. It's trending change, but you know, the data is the data. So this is kind of what we're looking at right now. And then lastly, we're getting into calcium signaling. So this is something also you can look at in the laboratory. And you can go to Wake Forest to do it. I will fly you to Wake Forest to do this. Dr. Vu and Dr. Marshall just came back from Wake Forest doing this work. So this is like hot off the presses. So basically, they looked at mitochondrial calcium uptake. So the highlight of this figure is basically, because I'm out of time, is basically when you knock out MICOS or MYC60 or CHCD6, you see a decrease in calcium uptake. And then we knocked it out using Western blots to see a small decrease because these are transient sRNAs. And then we also did this in liver, and so it's kind of the same thing. So I'll go through this pretty fast. So with liver, you do see these changes. The same thing with metabolism. But the cool part is you do see this robust change in MYC60 and OPA1, not only by transcripts, by Western blots. And so what we decided to do was actually knock these down, CHCD6 and MYC60, and you get a lot more robust deletion in this particular case. And so we're looking at now what's happening to ROS and other systems. Because the liver is something that you can challenge very easily, like doing fast refed conditions, or you can do caloric restriction or high-fat diet. We just finished cohorts of this with Yale, and then now what we're doing is actually going back to look at Western blots and ROS assays in the tissue to determine which particular challenge is needed to alter the system for MICOS complex. And then after that, we'll be done with this paper too. I'm so excited about this one. So this one's really cool because I wasn't expecting so much of a robust deletion. And it's from another lab too, so it's really nice to recapitulate what we have. And then I've kind of showed you a little bit about BioView, so I got curious if we could recapitulate some of these phenotypes. We looked at MICOS, there was nothing. But we said, well, why don't we look at, you know, some interactors of MICOS? So we looked at SAM50, which is part of this bridging complex that spans to the outer membrane. And one of those components is SAM50. And we found that SAM50 has been reported in a lot of different populations to be important for cirrhosis of the liver, non-alcoholic fatty liver disease. But in an admixture population, it hasn't been reported. So the South, everybody's very unhealthy. And so what we can do is get a lot of good clinical data showing this phenotype. And what we found was that this passes Bonferroni. So this is a different case. So when you have nominal significance like you did in the heart failure, it's less than 0.05. But Bonferroni is a very tight consideration. So it's the most stringent test in genetics. And because it passes this, it's at the cutoff line of like -5, but we go all the way up to -20, which is absolutely amazing. So we have strong confidence that we do see these associations. And so this is one of the reasons why we added the high-fat diet. And we're also looking at MERCs and then lipid droplets as well. So this is just preliminary. But I'm so excited about these two papers because they're like right on the cusp of being done. And you can join for the brain papers in spleen and sperm. And I want to thank my laboratory. This is a little outdated because there's a lot of people that I'm missing. But basically, these are the last 2 summer programs that we have. We actually have 15 new students for the summer, so it's really fun. They're all undergrads. I was scolded for having too many students this summer. Dr. Marshall runs all the money in the lab. So she was like, you got to find more money. So that's what I'm in the progress of doing. So basically, this is my funding. Of various sources. I look everywhere for funding. I just want to challenge you all as young investigators, the senior investigators know this, but you look everywhere. Don't spend your time only at the NIH or NSF or philanthropy, you look for nonprofit, you look for people that'll donate to you, you look for pharmaceutical companies. I look everywhere. I never am worried about how it looks to my colleagues. I just need to fund my lab. So sometimes I also will ask a big investigator, I will do all your EM and send my manpower to you if you give me an MOU. So like a memorandum of understanding, and they give me $100,000 and we do all that work. So I will find money in any kind of clever way to keep the lab doors open. So we're good for another 3 years right now, and then that's what I'm working on to do, that another round of showmanship in business. So thank you, and I want to thank my funders, and thank you for the opportunity to present. I've really enjoyed myself, and also thank you for training Dr. Edgar Garza-Lopez. He's been a great asset to the laboratory, and will continue to be. And so we hope that any of y'all will come to the laboratory. So thank you.
Thank you, Dr. Hinton. Before going to the questions, I would like to say something for the students. If someone wants to talk to Dr. Hinton, we can arrange a short conversation after the seminar. I mean, Dr. Hinton will be happy to talk to you. Well, I don't know if you have questions for Dr. Hinton. Andrea.
Great talk.
Thank you.
I'll just leave the questions. So you sampled mitochondrial structures in normal cases and in various disease conditions from various tissues. But if you look at the mitochondrial structures under the normal conditions, a lot of times there are cell type specific or tissue specific structures for the mitochondria. I wondered if you could comment on that. And then also, it seems that there are differential impacts to loss of certain MICOS genes that are also tied to tissue specificity. And I was wondering if you could speak on that as well.
Yeah. So it's a great question. So the first question, oh, it's loud, this is good. So the first question is around mitochondrial structural changes. And so one of the things that we're thinking about in the laboratory as a global hypothesis is that per tissue, the frequency and the organization of the tissue is different. So we have done, in collaboration with Brian Glancy, and he's published some by himself, in just regular skeletal muscle, at like a response to a gene like MICOS or OPA1. And in the cardiac tissue, there's more specialized like oval looking shapes that have a hole, the donut or the ring mitochondria, for lipid droplet interactions. And so we even see a higher frequency of those when the tissue has been starved or when mitochondria have been challenged in another way from like loss of like glucose but more fatty acid responses, and it's leaning towards another effect. The second question is about gene-specific responses. It's very true, and it's very controversial in the field. That's why we're doing it. I like a little drama, not too much though. So one of the things is that there is classically known in the field that MFN2 is this high-level gene that does a lot of different things. So it regulates fusion fission in the membrane of the inner mitochondria, but also ER contacts. But in some tissues, the distance doesn't always change consistently. So there was this big controversy about what do genes do specifically in a tissue. And in the eye, for example, OPA1 is very different. So in some of the other specialized cells, it forms certain contacts that are like ER lipid interaction contacts or vesicle contacts that are not what you normally see. So that's just an example of like why we have to study genes specifically in that tissue and take the context at one at a time. Like, okay, there's these organelles in this response. Then now in the system, we're learning that mitochondria don't communicate by themselves. And it's not even just with ER. Sometimes it can form a tri-complex with peroxisomes, if you will, to participate in beta oxidation, exhaustion of changes in calcium. So there's a lot of things that we're finding out about the organelle and how dynamic it is alone, but also in concert with other organelles. And so one of the things that we're getting towards the fall is to take the database to do the aging, but then challenge it with MICOS in those particular tissues, looking at Krelok's P system, and actually deciding like, is it specific to aging or is it specific to that gene and that response in that tissue? So that's one of the things that we're getting at. So great question. This is what we argue about, how fast we should go for this. This is why you bring talented people to the laboratory, right? So that they can challenge you to think differently. So great questions.
Excellent talk. There are so many things that I will take 1 month to process everything, but I have 2 questions anyway. One is very simple. Do you consider that mitochondria could be like a biomarker at some point for some kind of disease?
Yes, and I'm in the minority of that right now in the mitochondria field, but I do think that because they're specialized shapes. So usually mitochondria exist as a network, okay? They're not always separate, but when disease occurs, you see fragmentation, but no one describes the different types of fragmentation. One of the things I'm so interested in is because there was a paper that came out in Nature and one in Science that described the expression changes of certain factors that influence fission. So MFF is expressed highly in the midline of the mitochondria to participate in like proper fission, but FIS1 is expressed highly under stress or to do ER contact changes or other organelle changes, and it's on the end. So this made me think when I was writing that review paper, I was like, wait a minute, there might be different expression of proteins around the edge of the mitochondria per type, right? And so what we're hopefully doing next year is to figure out how to isolate the mitochondria in their native structure, then do mass spec imaging to look at their protein expression. So that is like my dream. So again, someone can come to do that. so you know, there's something that, you know, would be a very difficult project, but it would be higher impact than some of these other projects, right? So I'm it's a great question. It's where I'm thinking, and it would be really, really cool to be able to do that.
The other question, I'm a little bit ashamed because you talked about so many things, and this is not something you talked about, but that's why I'm making the question. Do you know if in stem cells during the processes of differentiation or de-differentiation or whatever, there are also these changes in mitochondria?
Yes, but I'm not supposed to speak specifically about this. So there's a collaborator that we have at Yale that looks at skin, and so she does see phenotypic and specific metabolism changes. So all I'll say is I don't want to steal her thunder because she's about to go for faculty, but the answer is yes, but I can't say how because I want to wait till her paper comes out. But yeah, but yeah.
So thank you for the wonderful talk. I was wondering if you would expect some mitochondrial changes to occur like only when there are specific lifestyles, or are there specific changes to aging that would happen to any type of, you know, patient or?
So that's a great question. There's a published paper, and our aging cell paper will be published. So there's a cell metabolism paper from Lucas Coriano that actually challenged the system where he did in young and old, but then gave old individuals that exercised a lot, gave biopsies, and then actually looked at gene expression changes. This gave me the idea, like, we should look at exercise. So there are some people that have looked at exercise by TEM and not 3D to show that the networks can rejoin, and they've taken biopsies and looked at it by confocal. We're in the process of writing a grant to NIA around actually resolving some of these structural networks with ATF4 mechanisms, so that's something we're interested in. We do have some preliminary data that suggests when you exercise, you can recover these transcripts, but in MICOS specifically. And we're waiting for readouts for MFN2, so that's ongoing in Brazil, so I'm excited. So.
Thank you.
First of all, thank you for your talk. I really love your enthusiasm. I really like your energy. But I'm really curious about how the shape of the mitochondria will change with the interaction with extracellular matrix. I mean, I know that mitochondria has like a really specific role with different type of ECM. Specifically with dystrophy, I know that sometimes if their type of detachment, it can lead to RICA2 and so on. But I'm curious that you like left those conversation out of your presentation. Like, I think that maybe some type of SSM might contribute with the changes that you are looking at, especially in the topics are related to skeletal muscles that you are able to see these wild things? I'm sorry, go ahead.
No, I'm laughing because Andre's behind you talking about, I told you. So we were at a conference last year in New Hampshire. Someone asked a question, and I was like, oh, not right now. And then I told her, I said, it's a cool idea, we'll do it later. So right now it's in a grant. So I had applied for HHMI. So if I get it, we'll do it because we'll have unlimited money to spend on it. But it's really expensive to actually not only clarify the matrix, but also change it. And so that's something that would take a lot of robust resources, so we're waiting to do that. But we do know that there's something in the hormone milieu that is responsible for changing interactions in metabolism, like when you're looking at platelets. That's from Dell's lab. So it begs the question about if you have hormones traveling everywhere, like it is circulating in these other tissues, so it's influenced the extracellular matrix. So that's a yes. And then it's a yes, and I wrote it in the HHMI grant. So hopefully we get it, and then we can do it.
Thank you.
Thank you. I also enjoyed your talk very much, very inspiring. Just two questions, a little bit technical. Is OPA1 also reduced in brain and in the eye retina? Is in aging, with aging?
It is. So that's projects that are coming up. So to tell you, so the brain is a lot more sophisticated. So we're looking at circuitry. So we're looking at the hypothalamus and amygdala. There is a paper out there that describes that with mtDNA mutations, there is altered in mitochondrial structure. That one's in the hippocampus. And so what we decided to do was do the amygdala and hypothalamus, which are understudied structures for mitochondrial metabolism. There have been some changes where you've knocked out OPA1. You do see this vacuolation in cristae morphology and a metabolic effect, specifically in POMC neurons. But at a global level, when we look at transcripts, just looking at the whole hypothalamus, we do see a decrease in gene expression, not only there in mice, but also in flies when you look at the whole head, and then also in cells. And then we've also, knocking out like neuro cells, you can see these change. But we also recently have done human tissue where we looked at qPCR of OPA1 and MICOS, and we see this phenotype. So now we're pursuing looking at the structural changes, and now looking at calcium signaling as a major kind of role in saying like, well, if MICOS is affecting calcium, what's the channel that it's associated with to do this? So that's why it takes so long, because the brain, to get a good paper like aging cell or redox biology or something like that, you need a lot more mechanism. And so we're having to collaborate a lot with the Tomar lab for its cell part, and Illinois and LSU to be able to do the calcium imaging for those two projects. And we hope to collaborate, which is why I'm also here to be able to do that in the future with Felix, to be able to look at like whole cell patch clamping, to look at like if we give the MICOS complex, how can we track it in like a whole cell bath and look at these changes. So there is some preliminary data to suggest yes, and that's the direction we're going.
Amazing. And one last question: the myofibrillar networks that you're finding, do you think they can be a compensatory effect in aging?
I think so. So this gets at the individual's question. I'm sorry, I didn't catch your name, but the individual's question around the extracellular matrix. So we do think that there is a role that it plays. So if it's receiving a signal, remember, I don't know if you've read the paper that when you delete OPA1, you see this myokine effect, like with GD50, typically FGF23, affects calcium signaling. So what we're thinking is that when you have this mitochondrial dysfunction or this shape description change, there's a myokine release that may be signaling to myofiber. So it's not just only you see the network hyperfuse and it influences it, but we also think there's some communication between this happening that we are just not familiar and don't know the answer to. So it's a question that's unknown for us right now, which is a great question.
Thank you, thank you.
Thank you.
Oh, yes.
Have you ever 3D profiled satellite cells from myofibers, either isolated myofibers or in sections? And if so, how different is that from fully developed muscle?
So the first answer is no. And the reason I laugh is because we started to do that in sperm. So we're trying to do the spermatogenesis. So we're doing it in axolotls and then doing it in mice and humans right now. And then if that goes well, then we will do satellite cells and kind of go from there. So it'll be, the different stages. You're a skeletal muscle expert, so you know. So, yeah.
Well, thank you very much.
You know, traditionally, the function of mitochondria has been related to metabolism. But recently, we know that they have also many things to do with inflammation. And I'm talking about the response against viral RNAs. And my question is if you think that there is a relationship between the shape of the mitochondria and the inflammation process in the tissues that you're looking at. And only because of your face, I know you know. It's something that I thought.
So I'm so excited because so we have a collaborator, Dr. Annette Carabo. She's like a very big deal in like heart world for American Heart and things like that. And she asked us, I felt so honored. I was like, yeah, we'll collaborate with you. So she has done this study where you do salt sensitive studies in humans, and she's done site-seq data, and she does immune cells. She looks at all the immune cell types. And so we see in monocytes specifically that there are changes in like glycolysis, just general metabolism. And now we're looking at MICOS, like as of like right now. We just like designed those projects. Our first collaborative paper will come out on cortisol metabolism shortly because we're about to submit it to hypertension. Because our joint student just got asked to submit it there. So I've been waiting on it. I've been telling them to come on. But the more interesting projects, in my opinion, are ATF4, GRP78, which is kind of just a general ER stress marker that control mitochondrial shape, and the MICOS complex, and the known master regulators of mitochondrial metabolism, because they're not well studied or captured in the immune cells. So what we're doing is knocking them out, and the immune cells that she has available that she isolates from humans, and then they'll be in suspension because they're hard to kind of like, like settle down. We'll then put them on an agarose gel to image them for like dynamics to see what happens after the gene deletion or overexpression, then do metabolomics and lipidomics, and then publish with kind of the correlation between blood pressure, gene expression, and then also like cortisol or other glucose related levels. So it's coming. So that's why I was smiling, because I was like, ooh, y'all are asking great questions. This is like stuff we're studying right now. Makes me so excited. So.
Thank you. And what do you think about the recent papers that shown they show some evidence about mitochondrial transfer between one cell and the other?
I knew you was going to ask that. I feel like this is being recorded. So we will say that it's a little controversial. It is. Oh, it's fine. You can keep it recorded. I'm just a little dramatic, so it's fine. So it's so I'm happy to defend myself anytime. I had to defend myself one time in front of the leader of the field. It was fun. So basically, there goes along with two things: mitochondrial transport and trafficking. So it is very interesting because we know that hemochannels are forming to where mitochondria are going through. There's a traffic paper that describes that. A lot of people haven't studied the mechanisms around how that happens. We also think that there's vesicles that engulf the mitochondria in a cell-to-cell communication. So we do believe mitochondria can traffic from cell to cell. And then we also do think that they can travel on the bloodstream. It's not very well established, but we do find loose mitochondria in the bloodstream. There was a FACET paper, and there's been 2 or 3 other papers out that are about that. And so right now, it is like, it's possible. Let's see some stronger evidence to back that up. It'll be really important. One of the things that I think that's really nice, especially on Twitter, the big people are there, and they will challenge your paper. They will let you know if it's good or not, if it's consistent with other data. So luckily, our papers have been consistent with other data. And so the reason I bring it up is because we actually got asked to speak a little bit about some of that work that we think around, like actually integrating mitochondria that have been refolded into other tissues. So we'll do that in Berlin in the fall. And so it's something that I think is controversial right now. I would like to be able to do that in a heart system where you can isolate, or in a skeletal muscle, give damaged mitochondria, let them either traffic on the portal vein somewhere to the site of interest, if there's like a receptor that could lock it in place as an option. Or the second thing that I'm thinking about is like just locally giving it to the tissue and seeing if it can traffic up, like if we can follow those with like GFP to see if they traffic not only to the source tissue but to other places. And so if they travel to other places, then we know that they're traveling by the bloodstream, because it would be hard for you to integrate from tissue all the way to like the liver. So these are things that we're thinking about. We haven't done those experiments, but it's something that we're in the process of hypothesizing. So great questions. So, any other questions? It's fine. I don't mind answering. I'm here all day. So.